fragments. In most animals or many animals, they are called as thrombocytes. Just like you have erythrocytes, leukocytes, you have the thrombocytes or platelets. And why they are called as platelets? Because they are not proper cells, but they are fragments. In human beings, they are fragments which are enucleated, and they are formed from very giant cells within the red bone marrow, which are called as megakaryocytes. They lack nuclei, but just like leukocytes, they can form pseudopodia, they can show phagocytosis, and they are capable of amoeboid movement. These platelets are born or they are synthesized from the precursor cells called as megakaryocytes within the red bone marrow, and they perform a very, very important function, which that is loss of blood. Whenever there is a damage to any blood vessel or there is any injury to the body or trauma to the body, they prevent blood loss, and they achieve by forming a very minute but very effective platelet plug, and thereby not only do they form a platelet plug and seal the blood vessel wall wherever there has been damage or injury, they also help in formation of a jelly like substance which is called as a clot. And once the wound has healed, they also help in the contraction of the clots. So these blood platelets, they are very minute, 2 to 4 micrometer in size or diameter. They are small, disc shaped, and they are found in the blood in a number. Just like RBCs are present in millions, WBCs are present in thousands, 5,000 to 10,000 per cubic mm of blood. These are present around 1.5 to 2.5 lakh to 4 lakh per microliter of blood. And these blood platelets, they are synthesized within the red bone marrow, and they are destroyed at the end of the lifespan within the by the macrophages which are present in the spleen and liver, and they can live or they survive up to 5 or 10 days, 9 days. 5 to 9 days is their regular lifespan. Now these discs which are pinched off from the megakaryocytes, they are nothing but they are being pinched off as cytoplasmic fragments. And within inside, they have got two different type of granules. They are very rich in granules. They have endoplasmic reticulum, they have granules, and these granules contain clotting factors and even certain growth factors which are derived from the platelets. And there are certain dense granules which are the specific granules which contain ADP, adenosine diphosphate, and adenosine triphosphate or ATP, calcium, serotonin, as well as FSF or fibrin stabilizing factor. So they are rich in the clotting factors, and that's how they promote or cause clotting of blood whenever there is injury and loss of blood. So here is a beautiful cartoon where you can see that they have got lot of ER, endoplasmic reticulum. These are the very large megakaryocytes or giant cells, and these giant cells, they have got lot of ER. And here you can see that in the megakaryocytes, they are produced by primitive stem cells, pluripotent or totipotent stem cells, which are called as hemocytoblasts within the red bone marrow by a process which is called as endomitosis, wherein large number of times the chromatin material divides or duplicates, the chromosome number duplicates, but there is no nuclear or cytoplasmic division, as a result of which in the nucleus large number of lobes are formed. And these lobes, the nuclei, the chromatin material, the genes, they contain information for synthesis of all the various type of clotting factors which are required for the clotting of blood. And as you can see from the megakaryocytes, by pinching off of the cytoplasm, small, very small disc-shaped, irregular disc-shaped granule containing structures are liberated. These are called as the platelets, and they help or promote clotting of blood. And the important molecule or hormone which is responsible for regulating the process of platelet synthesis is thrombopoietin, TPO. which is produced in our body by the liver and kidney. And this TPO regulates the synthesis of platelets from the by fragmentation of cytoplasm within the megakaryocytes, which undergo endomitotic division, that is DNA replication without any nuclear or cytoplasmic division. Each megakaryocyte can give rise to approximately on average 2 to 3,000 platelets, or we can say a general range is 1,000 to 5,000 of platelets. And these platelets contain at least 13 to 20 clotting factors which promote clotting of blood, and they also contain certain molecules which are very important to the process of clotting and platelet plug formation, and they include serotonin. And for every stage in the clotting process, there is a molecule which is required, which there is an ion, and this is calcium. Then there are certain enzymes, platelet growth factors, etc., which are required, and these are present within the granules of the platelets. Now, these platelets, they function by, they help in platelet plug formation by sticking together, and thereby they stem the flow of blood. They stem the loss of blood, that is, they stop the bleeding, and this process of arrest of bleeding or cessation or stoppage of bleeding, it is called as hemostasis. So now, with this kind of a background, let us go to the meat of the topic, the heart of the topic, which is clotting of blood. And the word clotting is also called as, alternative word is also coagulation. So let us look at in depth, let us study the mechanism of coagulation or clotting of blood. So what is blood coagulation? It is nothing but clotting of blood after damage to the blood vessels. Whenever the blood vessels get damaged, either due to minor or major injury, the body automatically, spontaneously responds by initiating the process of hemostasis to stop the clotting of blood. And this stoppage, it is referred to as hemostasis. Hem means blood and stasis means stable. Stasis means the blood becomes pooled at one place. Stable, it becomes collected at one place. And what is blood clot? What is clot? Clot is nothing but a gel. Normally in the blood vessels, the blood flows very freely and there is no obstruction or friction which they encounter. The blood never clots in intact blood vessels. But whenever there is friction or whenever the blood vessel is ruptured or damaged and the collagenous, underlying collagenous layer is exposed, then there is friction which is created and the blood becomes converted into a jelly-like substance. And this jelly-like substance is called as the clot. And initially this clot contains not only platelets, but it is stabilized by a network, a mesh-like network of threads. These are called as fibrin threads. And once the stable blood clot is formed, it traps the formed elements, that is both the RBCs, the erythrocytes and WBCs or the leukocytes. They become entangled within the fibrin threads. So now the next question arises, what is, when we allow the clot to stay for some period of time, the liquid portion, it separates from the clot and forms a fluid which is called as serum. So what is serum? Serum is nothing but blood minus the clotting proteins which have been used up in the clotting process. And very important clotting proteins are fibrinogen, prothrombin, which are present as a precursor molecule within the blood. So once these clotting proteins have been used up to form the clot, whatever blood, the liquid portion that remains behind, it is serum. It is called as serum. Serum is nothing but plasma, the blood minus the clotting proteins. Now the question arises, what is the process? How does this blood clot? Is it a very simple process or is it a complex process? It is a very complex process. It is not a simple process. It uses a lot of clotting factors, and these clotting factors are already present within our blood in an inactive state. So now initially for the clotting cascade to be activated, The first thing is the aim is to stop whenever the blood vessel has been subjected to injury or trauma. The first aim is to arrest the bleeding of blood or produce hemostasis. And in this process of hemostasis, a number of events occur which prevent excessive loss of blood from the damaged blood vessels. And there are three important events, or hemostasis takes place by three important steps. The initial most earliest step, which is very prompt, which occurs very rapidly within seconds, is the reflex vasoconstriction or vascular spasm. That means by spasmodic contraction, vasoconstriction take place. The damaged blood vessels, they constrict, they contract so as to, just allow the rupture to become narrow, so that excessive loss of blood is not there. So reflex vasoconstriction of damaged blood vessels to reduce the amount of loss or to completely stop the loss of blood, it is called as vascular spasm. And this occurs due to contraction of the smooth muscle wall. The blood vessels, they are ones in which the blood flows, and internally they have a layer which is in direct contact, epithelial layer which is in direct contact with blood. This is called as the endothelium. Next to the endothelium is there is the smooth muscle wall. So whenever the blood endothelial layer and the collagen layer rupture within the blood vessel, the blood starts to leak out. The smooth muscle vessel wall also leaks, ruptures. Then the blood starts to leak out. And when the blood starts to leak out from the damaged or injured blood vessel, at that point or site of injury, lot of platelets are attracted, and they try to come and seal. See, while the vasoconstriction is already initiated, the platelets are also attracted to the site of the injury by the chemicals which are released by the damaged platelets, and they come and they adhere or attach themselves to the blood vessel wall at the site of injury, and this they form what is called as a platelet plug. This is called as the primary hemostatic plug. This is a relatively unstable plug, and this plug is stabilized by the formation of a mesh or network of fibrin threads. And this then it is called as the stable hemostatic plug or the secondary platelet plug. And this plug is formed, the stable plug is formed once the clotting cascade or the mechanism of coagulation or clotting of blood is initiated, involving large number of clotting factors. So thus clotting of blood has got three important steps or events which take place. One is reflex vasoconstriction by vascular spasm within the blood vessels, damaged blood vessels. The second is the platelet plug formation, which is the primary platelet plug, which seals off. And there are chemicals like serotonin, etc., which are released by the damaged platelets, the granules within the platelets, and they are responsible for attracting the platelets to the site of injury to form the platelet plug. And the third is initiation of the clotting cascade, which results in the formation of the secondary hemostatic platelet plug, which is the actual blood clot, which is the true blood clot. So let us look what happens. Let us study in detail what happens during vascular spasm whenever the blood vessels are damaged. Here you can see, this is the innermost layer. This layer here, you have the endothelial cells. All of these cells here are the endothelial cells. These are the RBCs. These blue ones or purple ones are the platelets. So initially, whenever there is a vessel injury, the blood starts leaking out or blood starts flowing out. And then what happens, that these smooth muscles of the blood vessels, they undergo reflex vasoconstriction. Now this is very important. Otherwise, you can just imagine a piece of cloth or any dress which you are wearing, it gets torn. Torn means there is a slit. So whenever there is damage to the blood vessel also, there are the muscles, the collagen layer becomes exposed, the muscles rupture, and the blood starts oozing out. So what we do whenever our dress is split up into two parts, or whether when it is torn, we try to pull both the edges together to narrow. And then what we do, we take a sewing thread and needle and we try to seal that damage. Okay, we try to seal the opening. Same way here also, those events are initiated naturally, spontaneously. Whenever any blood vessel damage is there, the smooth muscles within the wall of the damaged vessels, they contract, and this reaction or this process is called as vascular spasm, and this reduces the blood loss for a certain period of time, maybe a few minutes, during which the other mechanisms, hemostatic mechanisms, come into play, resulting in formation of the primary platelet plug, and finally initiate the clotting cascade, leading to the formation of the actual blood clot. So this spasm is caused by damage to smooth muscles, by substances which are releases by degranulation of the damaged platelets, and also by reflexes which are initiated by pain receptors which are innervating the blood vessels. Now, how is the platelet plug formed? The platelets, they are become, damaged platelets become very sticky, and they come and adhere to the broken part of the collagen fibers and the damaged endothelium. So these platelets come and accumulate over here wherever there has been damage to the blood vessel. They come and seal that damage by sticking to it, and this process is called as platelet adhesion. Now, the second step in platelet plug formation is platelet release reaction. Once they become adhere to the platelets adhere to the wall of the damaged blood vessel, they become activated, and their characteristics also start changing. They form numerous protoplasmic extensions or projections, which are called as pseudopodia. They start interacting with each other. The platelets start interacting with each other. They become very sticky, and they initiate a step which is called as release reaction, whereby different chemicals like ADP, serotonin, etc., thromboxane A2, all of these are liberated from the damaged blood vessels. See here, these damaged platelets, they start liberating these chemicals into the surroundings, ADP, serotonin, thromboxane A2, and this response, it is called as platelet release reaction. The last step in platelet plug formation, primary platelet plug formation, is the release of ADP. It makes all the platelets sticky. They start also sticking to each other by contacting each other by means of their pseudopodia, and they gather together, and this step is called as platelet aggregation. They accumulate, they gather together to seal. This is the opening which has been created. They seal this opening. So the platelet plug formation involves platelet adhesion to the damaged blood vessel, followed by release of certain chemicals like serotonin, ADP, etc., making them sticky, and they aggregate to each other, and they eventually form a primary hemostatic plug, which is called as the primary platelet plug. So reflex vasoconstriction, primary platelet plug formation, while these are occurring, the third step which takes place is the initiation of a amplification cascade. Amplification cascade, which is called as it's a complex mechanism of clotting of blood, whereby the actual gel-like clot or is formed within the blood, and the process of this formation of this jelly-like clot is called as clotting or coagulation. It is a cascade of a number of enzymatic reactions or chemical reactions which results in conversion of an inactive protein which is present in our blood called as fibrinogen. That gen itself says that it is a precursor protein. The fibrinogen which is already present in the blood is converted into fibrin threads. Initially, these fibrin threads are in the form of monomers, and then they polymerize. And once they join to each other, link with each other, polymerize,
Hello, Seema ma'am. Hello? 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 Hello, Seema ma'am. Hello, Seema ma'am. Hello? Hello? Hello, Seema ma'am.
Hello, sir. Hello, sir. So let's start with the clotting process of clotting of blood or coagulation of blood. What is blood coagulation? It is nothing but clotting of blood once the damage has occurred to any injury has occurred to any blood vessel. When our blood vessel is cut or damaged or ruptured, or whenever there is accident, or even during surgery, the clotting process is initiated. Coagulation of blood is initiated whenever there is damage to the blood vessel. It is called as hemostasis. Hemostasis is the process whereby the blood hemostasis is the process by whereby the blood is the clotting of blood is initiated and the leakage of blood from the damaged blood vessel is arrested. There is stoppage of bleeding. Now, what is the blood clot? It is a jelly-like substance, a gel, which contains formed elements of blood which are entangled within network of fibers made up of fibrin. So how does this blood clot? It clots by a process which is called as hemostasis.
Hello, ma'am. Hello.
It is a positive feedback mechanism and a large number of clotting factors. There are around 30 clotting factors which are involved, which lead to formation of clot within the blood. So clotting of blood includes three important events: vascular spasm, platelet plug formation. This is an unstable primary platelet plug. And finally, there is an actual blood clot which is formed as a result of formation of fibrin threads which seal the platelet plug. Now, vascular spasm, whenever the blood vessels are damaged, the blood leaks out. Whenever the blood vessels are damaged, the blood leaks out at the site of injury. But
मैम
at yes,
आपको
yes,
पीपीटी शेयर नहीं हुई है मैम. हेलो.
yes, sir.
आपका स्क्रीन शेयर कीजिए ना.
हाँ किया है सर. Again, I will share. अभी मेरा स्क्रीन आप देख पा रहे हैं?
Yes.
Can you see my presentation?
Yeah.
Okay, sir. Sir, the moment you see there is a problem, please tell me. Thank you, sir.
Okay.
So, how does the blood clot? How is the blood clot formed? Blood clot is formed by the process of hemostasis. In it, this hemostasis means arrest of bleeding at the site of damage to or trauma. Wherever there is mechanical or physical trauma, there are a number of events which take place which to curtail the blood loss, and there are three important events which occur: reflex vasoconstriction of the damaged blood vessel to reduce the blood leakage, then formation attraction of platelets to the site of injury, and formation of the primary but unstable platelet plug formation, and last but not the least, coagulation or clotting of blood. And this process is the most complex process which involves at least 30 clotting factors, out of which there are a number of 12 important clotting factors, and they were they were provided Roman numerals as numbers in order of their discovery. They promote, they are called as procoagulants, which promote the clotting of blood. And clotting of blood thus involves three important events. One is the reflex vasoconstriction by contraction of the smooth muscles and they initiate what we call as vascular spasm. Then second is formation of a platelet plug. This is the primary platelet plug, and this is an unstable platelet plug which is stabilized by formation of the actual blood clot, which involves fibrin network of fibrin fibers. Now, vascular spasm whenever there is a blood vessel injury, when the blood vessels are damaged, the smooth muscles of the present in the wall of the blood vessels, they contract. So the blood, whenever there is vessel injury, the blood starts coming out of the damaged blood vessels. Now, how to prevent or how to stop that? So the smooth muscles involuntarily reflex vasoconstriction is there by vascular spasm, and this reduces loss of blood from the damaged area for a certain period of time, maybe a few minutes, so that the other two important hemostatic mechanisms will come into play. Then the second is platelet plug formation. So what this platelet plug formation takes place in three steps. One is attachment of platelets to the broken wall of the blood vessel, where the platelets come and contact the damaged blood vessel. This process is called as platelet adhesion. This is followed by platelet release reaction, in which the platelets, they are rich in granules containing the clotting factors. So these platelets at the site of injury, they get damaged, they become ruptured, and there is release of the clotting factors and chemicals like serotonin, ADP, etc. from their granules. And the liberated ADP, serotonin, and thromboxane A2, they make the platelets very sticky, and these platelets, they accumulate over there, they aggregate, accumulate, and they form a stable, they form a primary platelet plug. Sorry, it is unstable. It is a primary hemostatic plug or the platelet plug. So the platelet plug formation initially takes place by attachment of platelets, platelet adhesion, followed by degranulation from the damaged platelets. Many chemicals like serotonin, ADP, etc. are liberated, and these cause the platelets to become sticky. They come and adhere, attach to the, and they form a plug at the, they form pseudopodia. They contact each other, and they form a platelet plug at the site of damage to the blood vessel. And lastly, once this platelet plug is formed, simultaneously, the last and most important step in this mechanism of hemostasis is the coagulation cascade which occurs. This is a blood clotting or coagulation mechanism, and it results in formation of a jelly-like substance at the site of injury, which is called as the blood clot. And it is nothing but a series of number of enzymatic reactions or chemical reactions that take place, resulting in formation or conversion of fibrinogen, which is a protein which is present in our blood in an inactive condition, fibrinogen, and it is responsible for formation of fibrin threads. Now these fibrin threads, they polymerize and form a network, mesh-like network, to seal the opening, and this opening, this process, it is called as clotting. And this entire the mesh of fibrin fiber, network of fibrin fibers, in which are trapped the platelets, so it is what it is doing, it is consolidating the primary platelet plug, and it also traps RBCs, WBCs, etc. at the site of vessel injury, and thereby it forms a stable secondary hemostatic plug. Okay, so clotting involves several substances which are called as coagulation factors or clotting factors. Now, what are the very important steps which occur in this clotting cascade? As I told you earlier, it is a positive feedback mechanism. This is a very important example. There are very limited or few, most of the control mechanisms or systems in our body are negative feedback mechanisms. This is a classic example of a positive feedback mechanism, in which one step, it amplifies and promotes the next step and keeps on amplifying it. That means it is an amplification cascade, and it involves large number of clotting factors. but there are 3 to 4 major steps which occur in the clotting cascade, and that result in the formation of the fibrin network. So the very first step, in our blood are present certain plasma proteins. You know, there are 3 type of plasma proteins. The first is albumin, large number, smallest but largest in number. Then you have globulins, and lastly you have the clotting protein, which is called as fibrinogen. And there is another very important clotting protein which is present in our plasma, which is prothrombin. Both of them are present normally, usually in our blood vessels, and they are present in an inactive condition, and they do not promote clotting of blood. Only when there is tissue trauma or when there is damage to our blood vessels, there is injury to our blood vessels, both by 2 different pathways. There are 2 pathways, extrinsic as well as intrinsic pathway. Prothrombin, which is present in an inactive condition. Pro mean precursor molecule. Thrombin means the protein, the plasma protein. So the prothrombin, which is present in an inactive form, it is becomes now activated and it is converted into thrombin. So the question you will ask, how is prothrombin converted into thrombin? There is a molecule which is formed, which is not present in our body. This is called as prothrombin activator. It is an enzyme, and its technically correct name is prothrombinase. Now, what does this prothrombinase do? It converts prothrombin into thrombin. And once this thrombin is formed, it converts fibrinogen, the other protein which is present, clotting protein which is present in our blood, and it is also a precursor molecule. It converts fibrinogen into fibrin. Please understand that in our intact blood vessels, blood never clots. There is no force or there is no reason for any friction. And there are certain molecules, prostaglandins like prostacyclins, they are coating our endothelium of the intact blood vessel. So the blood does not clot in intact blood vessel. If it clots in intact blood vessels and the blood flow is arrested, then it may cause heart attacks if it occurs in the coronary arteries of the heart, or if it occurs in the arteries or in the brain, then it leads to brain attack or what we call as stroke. And then the clot which forms in intact blood vessels, it is called as thrombus. And the clot, when it sometimes the pressure of the blood, flowing blood, may dislodge the clot, which may be tiny, and it may travel and go to vital organs like heart and get lodged in the heart or the lung, causing pulmonary embolism in the lung, or it may become enlarged in the brain. Then it may cause brain attack or stroke. So clotting within the normal intact blood vessels is not a normal process, and it is a medical, it may lead to medical emergencies like heart attack, strokes, pulmonary embolism. So normally in intact blood vessels, blood will not clot. Whenever there is either tissue trauma or there is blood vessel damage, then either by the extrinsic or the intrinsic pathway, prothrombin activator will be formed, which is nothing but a combination of activated factor number 10 and 5, and in the presence of calcium. Calcium is a very, very important ion. It is required at every stage of the clotting process. And then prothrombin, which is present in our blood in a precursor form, it is converted by prothrombin activator into thrombin. Then this thrombin converts fibrinogen into fibrin monomers. These fibrin monomers are still soluble. They're small threads. Then they form a mesh or network, and they polymerize by polymerization, and then they become insoluble. And this clot is stabilized by a factor which is called as the fibrin stabilizing factor, or factor number 13. As you can see, all these factors have got Roman numerals, and these factors were numbered as per the order of their injury. So just to review or summarize the important steps in the clotting cascade, either by extrinsic or intrinsic pathway, first of all, the prothrombin activator, also called as prothrombinase, is formed. Then this prothrombin activator, it converts prothrombin already present in the blood in an inactive state to the thrombin protein. Then this thrombin protein converts fibrinogen into fibrin monomers, and lastly the fibrin monomers polymerize, become insoluble, and they form a mesh-like network which is called as the stable clot, and within this stable clot are trapped all the platelets. So what does this fibrin clot do, mesh do? It consolidates the primary unstable hemostatic plug which was formed by the platelets. So you know, whenever there is a rupture in your dress or in a cloth, then what we try to do, if there is a slit or a rupture in the cloth, if the cloth, a piece of cloth is torn, we try to stretch or pull the two torn pieces together to try to seal or reduce the torn area, exposed area. And then finally we take a needle and thread and then we sew the broken parts or torn parts of the cloth. Same way here, initially the platelets are coming to seal the broken, damaged part of the blood vessel, and later on the fibrin network is being formed, which will form that mesh, which is equivalent to a stitching the torn pieces of a clot together, and thereby forming a stable hemostatic blood. And for the formation of the first step, the prothrombin activator, the formation of prothrombin activator or prothrombinase may be initiated either by tissue trauma or by damage to blood vessels or by both. So there are two pathways, extrinsic as well as intrinsic pathways, which are responsible. And as over here in this diagram, you can see that these are the fibrin, these are the fibrin fibers forming the mesh, the yellow ones. Now, before I actually move to the extrinsic and intrinsic pathway, let us discuss the different factors which are involved in the clotting of blood. As I told you, there are many factors, 20 to 30 important factors. But the very important factors which are actually involved in the clotting cascade, which is an amplification cascade, that means few molecules of one factor become activated. Each molecule then activates by in its turn. It enrolls more molecules of the second factor. And then they are become activated. So each clotting factor which is present in our blood is in an inactive state. A small oligopeptide or small unit is cleaved off. Active site is exposed. It becomes activated. It activates, and each such molecule then activates the next molecule in the pathway or the next factor in the pathway. So this is a positive feedback mechanism, and it is an amplification cascade, and thereby the process becomes very, very rapid, and it is required to be rapid. It is important, it is very important that the injury or the leak should be sealed off, and excessive hemorrhage or loss of blood should not take place. So first, let's have a overview of all the clotting factors. Most of these clotting factors are either present in the platelets or they are synthesized by the liver, and some of them are involved in the extrinsic pathway, some of them are involved in the intrinsic pathway for formation of prothrombin activator, or some of them are required and are formed in the common pathway, that is prothrombin to thrombin and fibrinogen to fibrin. So let's look at the different factors and the numbers that they were given. They were given Roman numerals as numbers, and these numbers were given as per their order of discovery. As you know, in the blood already fibrinogen and prothrombin are present in an inactive state, so very readily and discovered first. So fibrinogen was factor number 1. Factor number 2 is prothrombin. Then tissue trauma leads to the formation of tissue factor or thromboplastin. It is called as factor number 3. Then at every step I told you that calcium is required for clotting of blood, and that is factor number 4. Now we come to special names and the special factors. Factor 5, factor 7, factor 8, factor 9, factor 10, factor 11, 12 and 13. 13, as already told you, involved in the last step. It is called as fibrin stabilizing factor of FSF. As you can see over here, there is no factor number 6. This is because this factor number 6, initially there was a factor which was called as factor number 6, but then they realized that this is not present in our blood. It is formed at the time of clotting cascade, and it is nothing but amalgamation or combination of activated factor number 10 and factor number 5, and this is called as the prothrombin activator. So you do not see any factor number 6. Now these are very important questions which may be asked during the exam. Give an account of the mechanism of clotting of blood, give an account of the different clotting factors. So you have to explain the names of the different clotting factors, where they are synthesized, what is the role of each clotting factor within the clotting cascade. So the first clotting factor is fibrinogen, second one prothrombin, third is tissue factor or thromboplastin, fourth one calcium ions, and they are involved in all the steps, important for all the steps of the clotting cascade. Now let us come from factor number 5 onwards. Factor number 5 is called as proaccelerin, or it is also called as the labile factor. Now proaccelerin means it promotes, it accelerates the process of clotting, and it accelerates the process of formation of, it is a labile factor because it is used up, it is used up in the reaction. It combines with factor number 10, activated factor 10, and forms a prothrombin activator. Therefore it is called as the labile factor. It is very important because that is the starting point, that is the starting point of both the extrinsic as well as the intrinsic pathways. Now factor number 7, there is no factor number 6. Factor number 7 is called as serum prothrombin conversion accelerator, or it is also called as stable factor. So in contrast to factor number 5, which is used up labile factor, you have factor number 7, which is a stable factor. And then factor number 8, now it is easy to remember, you will be finding it easy to remember it in this way. Factor number 8 is anti-hemophilic factor. You know, hemophilia is a disease which is responsible for delayed clotting of blood, and it occurs due to deficiency of a factor number 8. And this factor number 8 is therefore known as anti-hemophilic factor or anti-hemophilia factor A. It is, its deficiency leads to it causes the classical hemophilia or hemophilia A. Then there is a factor number 9. It is also called as Christmas factor, or it is also called as anti-hemophilic factor B. So Christmas factor or Christmas disease is deficiency hemophilia it is also called as hemophilia B. So deficiency of factor number 9 causes Christmas disease or Christmas disease, or it is also called as hemophilia B. Then you have factor number 10. 10 is called as the Stewart-Prower factor, or it is also called as not prothrombinase. Please note, it is called as thrombokinase. It is thrombokinase. Then factor number 11. Factor number 11 is called as anti-hemophilic factor C. Its deficiency is responsible for causing hemophilia C. And factor number 12 is called as Hegman factor, glass factor, contact factor, or anti-hemophilic D. So it will be easy for you to remember it this way. Factor number 5 used up in the reaction called as labile factor. Factor number 7 is labile stable factor. Then factor number 8 is anti-hemophilic A. Factor number 9 is anti-hemophilic factor B, or anti-hemophilic factor B is also called as is responsible for Christmas disease. Deficiency is responsible for Christmas disease, so it's also called as Christmas factor. Factor number 10 is called as Stewart-Prower factor or thrombokinase. It is important for formation of prothrombinase. It combines with factor number 5, activated 10 combines with 5 and forms prothrombin activator. Then factor number 11 and 12 are again easy to remember, anti-hemophilic C and anti-hemophilic factor D. Lastly, you have the fibrin stabilizing factor, which is also called as clotting factor 13. So most of these factors, as you can see, are formed in the liver, but it is tissue factor which is formed by damaged tissues as well as by damaged platelets. The calcium is a part of our diet. It is also found in the bones, and calcium is also produced or synthesized within the platelets. So, and it is required, if you look at this table, it is required at all steps. That means whether it is the extrinsic pathway, intrinsic pathway, common pathway, it is required at all steps or in all the stages or phases of clotting cascade. Now, factor number 5 is produced by the liver and platelets. It is because factor number 5 is labile factor. It will combine with activated factor 10A and then form the prothrombin activator. So it is important in both extrinsic as well as intrinsic pathways. And rest of all the factors are synthesized by the liver, and also fibrin stabilizing factor is synthesized by platelets. Now you can understand better whether the role in either the extrinsic, intrinsic or common pathway when we actually look in at detail the role of different clotting factors involved in both the intrinsic as well as extrinsic pathways and the common pathway. So there are, as I told you, there are three first very important stages. So what are the first stage? Formation of prothrombin activator. It may be formed either by the intrinsic pathway or by the extrinsic pathway. So here, once this prothrombin activator is formed by combination of activated factor number 10 and factor number 5 together, then is initiated the common pathway. That is conversion of prothrombin to thrombin, and thrombin then promotes the conversion of fibrinogen into fibrin, and fibrin, it is it is responsible for formation of it gets the fibrin monomers get polymerized. They become cross-linked with each other in the presence of calcium, and also stabilized by activated factor number 13, also called as fibrin stabilizing factor, to form the actual cross-linked fibrin network or mesh or the clot. Okay, so let us look at the common pathway. I hope is very clear. Now let us look in detail at the various steps involved in both the intrinsic and extrinsic pathway. Let me first start with the extrinsic pathway. This pathway is initiated whenever there is tissue damage and the membranes, phospholipids, etc. are exposed, and whenever the which factor is released, this factor is called as factor number 3 or tissue factor or thromboplastin. Then what it does, it activates factor number 7. So tissue factor 3 and activated factor number 7 together are responsible for activating factor number 10. Now this activated factor number 10 then will combine with factor number 5 in the presence of calcium, and here PL stands for phospholipids, and convert prothrombin to thrombin. Thrombin will convert fibrinogen to fibrin. Fibrin monomers will then polymerize to form the fibrin network. Okay, so there are, which are the important clotting factors involved? Clotting factor number 3 in the extrinsic pathway. It activates factor number 7, and this 7 is converted into 7A. This 7A, activated A, it combine, and in the presence of calcium. It's very important. Out all these steps, calcium is required. So what it does is it activates factor number 10, converts it into 10A, and then this 10A will combine with factor number 5. So 3, 7, they are 3, 7, 10 and 5 are important for the extrinsic pathway. Now let's look at the clotting, all the rest of the clotting factors other than these 3, 7 and 3 and 7 are involved in the intrinsic pathway. Again, very easy to remember. You go in the reverse order. Factor number 12, whenever there is blood vessel damage, the collagen is damaged. The collagen layer is within it's a connective tissue layer of the blood vessel is damaged. It is exposed, and what it does, all these factors are present in an inactive condition in the blood. It activates factor number 12. The activated factor 12 then converts factor number 11. It activates factor number 11. Then this 11, again calcium is required at every step. Then this 11 activates factor number 9, converts it into active 9, and this active 9 in the presence of factor number 8 as well as calcium is responsible for converting. Just like factor number 3 and 7 were important for converting factor number 10 to active factor number 10 in the extrinsic pathway, factor number 8 and 9 convert 10 into 10A, and the rest of the common cascade is then activated. So at every step here, you will see that calcium and phospholipids are again, phospholipids are present in the plasma membrane. They are present in the organelle membranes. They are present in the RBC membrane. They are present in the platelet membrane. So the unit membrane structure is there covering the platelets also. So exposed phospholipids are a very important clot. They are promote clotting of blood in the presence of calcium. So again, to repeat, which are the factors involved in clotting cascade? Factor extrinsic pathway for involves factor number 3, factor number 7, factor number 10, and which combine activated factor 10 combines with factor number 5 to form the prothrombin activator. Now let us look at the intrinsic pathway. It involves which factors? Factor 12, factor 11, factor 9, factor 8, and of course factor number 10 and factor 5. These are again very important factors which promote formation of prothrombin activator via the intrinsic pathway. Then once the prothrombin activator, that is a combination of factor 10, activated factor 10 and factor 5 is formed, then it's an enzyme. It converts prothrombin to thrombin in the presence of calcium and phospholipids. Then this thrombin activates fibrinogen, converts it into fibrin. These are initially monomers. They cross-link with each other. They polymerize in the presence of calcium and they form the fibrin network, which is stabilized by the presence of activated factor number 13. Then because of which it is also called as fibrin stabilizing factor. So now let's go back to this, and as you can see, fibrinogen is involved in the common pathway. Prothrombin is also involved in the common pathway. The tissue factor or thromboplastin, by the name itself, you understand tissue factor mean intrinsic pathway. Sorry, extrinsic pathway. So it has a very important role to initiate the extrinsic pathway. Calcium and ions are involved at every step, and therefore they are involved in all the pathways. Then labile factor is used up both in extrinsic and intrinsic pathways. It combines with factor number 10, and factor number 10 therefore is again important both in extrinsic as well as intrinsic pathway over here. Whereas factor number 8, 9, 11 and 12, all of them are involved in the intrinsic pathway, whereas factor number 7 and factor number 3 are important to the extrinsic pathway. Then fibrin stabilizing factor, it is common to both the pathways. Now once the clot has been, I hope I have made it very clear. Now once the clot has been formed, as you know, very well know, or as I had explained earlier, I don't know, I lost you in the middle of the session, that once the jelly-like clot has been formed and you keep the blood sample for a short period of time, then the fluid part, yellow colored fluid part separates out. And this is so plasma minus the clotting proteins like prothrombin, fibrinogen, etc., which have been used up in the clotting process, that is called as serum. So you have to be very familiar with all these important terms. Blood, how it is different from plasma, how it is different from serum. Serum means that blood sample in which the clotting factors have been used up. So it is plasma or blood sample minus the clotting factors. So it is called as, it's not the plasma now, it is called as serum. And which are the formed elements? RBC, WBC, and the enucleated or lacking a nucleus, the platelets, disc-shaped platelets. So platelets have a very important role in stopping hemorrhage or arresting the bleeding or hemostasis by initiating the clotting cascade. And one thing which is very important is for you to understand that this is a positive feedback mechanism. It is an amplification cascade whereby one factor activates large number of recruits, large number of molecules, and each factor and thereby speeds up the process so that the process is very rapid. So it is a positive feedback mechanism. It is an amplification cascade. So whatever factor 10 is activated, it provides positive feedback on factor number 7, and it initiates also positive feedback Thrombin also provides positive feedback and initiates the intrinsic pathway, or it activates the intrinsic pathway.
हेलो हेलो मैम हेलो हेलो मैम हेलो 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 मैम हेलो हाँ मैम, आपका फिर से स्टॉप हुआ है। हेलो, आपका वॉइस फिर से स्टॉप हुआ है, वॉइस और वीडियो। हेलो, हाँ, ओके।
I have rejoined. I am sorry, there are some connectivity issues.
Okay.
Sir, which was the slide on which I got disconnected?
Your slide, वो जो ब्लड सर्कुलेशन बता रहे थे ना आप?
Clotting mechanism, sir.
हाँ, स्लॉटिंग मैकेनिज्म। 19, slide number 19.
Okay, thank you so much, sir.
Okay. Not this
So
one.
to sum up, not this one.
Not. Next.
Which one, sir? This
हाँ,
one.
यही था।
Thank you, sir. So once the clot is formed, and it has outlived its utility, then gradually the clot stops, the injury starts healing, and the clot stops dissolution. It stops, the clotting process stops completely, and the clot is dissolved or it disappears. And this process occurs because of the action of thrombin on an inactive molecule, or which is present, precursor protein which is present in our blood, which is called as plasminogen. Now this plasminogen is converted by thrombin and another factor which is called as TPA, tissue plasminogen activator. It is converted into plasmin. Small role is also there of lysosomal enzymes. And what this plasmin does is, It dissolves the fibrin clots or the fibrin fragment. It hydrolyzes them, it breaks them down so that the clot will then finally disappear and the injury can heal. So the clot which is dissolved by activity of plasmin, an enzyme which hydrolyzes the fibrin net fragments. Now, as I told you before, that normally blood should never ever clot in intact blood vessels. It should flow smoothly through the veins, arteries, and capillaries. But it has been seen that more problems are there in the body, or our health issues may be created due to it is more dangerous for blood to clot in intact blood vessels, because then it leads to thromboembolic disorders. Thrombus is a stationary blood clot in an intact blood vessel, whereas embolus, when the blood flows and the pressure of the blood may dislodge the thrombus. The thrombi get dislodged and they move and migrate to maybe other organs like heart, causing heart attack, or going to the brain, causing brain attack or stroke, or even lodging inside the lungs, causing pulmonary embolism. So all of these thromboembolic disorders, they lead to many of our health issues, cardiovascular problems, heart attack, stroke, etc. Okay. Now, I just wanted to make it very clear that danger is very less because of failure of clotting mechanism as compared to cardiovascular problems, which are very important lifestyle disorders today, heart attack, stroke, etc. So let's come to the last topic in our in today's lecture. What happens whenever the blood fails to clot? Now, what could be the reasons for the blood to fail to clot? Maybe the number of thrombocytes are very few in the blood. Normally we need, sir, am I still connected? Sir?
Yeah, ma'am, are you connected?
Okay, I'll continue. So normally what happens is we have around 2.5 to 4 lakh platelets in a per cubic milliliter of blood. But at least 1.5 should be there. So if the number falls below that, then it may create some or the other problem, bleeding disorder. Okay. And so platelet deficiency could be one of the reasons. You know, in dengue, that is a fever, dengue fever, it happens, it's a viral infection. And this in this particular condition, the number of platelets become very low, 60, 70,000. It may prove fatal also. So one of the clotting coagulation defects may be due to either lack of platelets, appropriate number of platelets, or decrease in the number of circulating platelets, or there may be some structural defects in the platelets. Then that also can lead to platelet defects. So failure of clotting mechanism will lead to bleeding disorders, abnormal bleeding, and it may be caused, one, by deficiency of platelets. Deficiency of platelets is called as thrombocytopenia. Thrombocytes is equivalent to platelet, and thrombocytopenia means deficiency of platelets or abnormally low level of platelets. Usually when the number falls below 80,000 or 50, 60,000 per cubic milliliter of blood, per cubic microliter of blood, then it is called as thrombocytopenia. And sometimes autoimmune disorders, antibodies may be produced against the platelets. So platelets may be being produced in the blood. They may be produced in sufficient number, but due to autoimmune disorders, the platelets may be destroyed by the antibodies. Another reason would be lack or deficiency of different clotting factors. It is deficiency or coagulation defects or of the clotting factors. For a very classic example is hemophilia. Hemophilia is delayed clotting of blood because of deficiency of different clotting factors. For example, classical hemophilia is occurs due to deficiency of factor number 8, and this can be treated because today in the market, as a result of recombinant DNA technology, by recombinant DNA technology, factor 8 can be synthesized, and factor 8 factor 8 can be administered. So if there is a thrombocytopenia or platelet deficiency, one can give platelets, or rather bone marrow transplant or blood transfusion can provide the missing platelets. Of course, compatibility and everything is again very important. Then coagulation defects, one can provide factor 8 concentrate in case of hemophilia, or Christmas factor 9 concentrate can be provided. Then I already discussed thrombus and embolus. Thrombus is an abnormal clot which develops in a intact blood vessel. It's a stationary blood clot. In contrast, traveling blood clots, they are called as embolus. Free thrombic clots which are carried via the blood, they flow in the peripheral circulation and they may get trapped within the arterioles, within the brain, kidney, lungs, etc., leading to heart attack, stroke, and pulmonary embolism. Now, let us look at this table in which I have discussed about different factors other than thrombocytopenia, that is deficiency of platelets. Let us discuss various coagulation defects which are responsible for bleeding disorders or failure of the clotting mechanism to take place. Now, factor number 1 is a very vital factor, fibrinogen. Now, if there is deficiency of fibrinogen, which is called as hypofibrinogenemia, or the complete absence of fibrinogen, it is called as afibrinogenemia. So these are rare congenital disorders. That means they are present right from birth. And this deficiency, of course, if there is no fibrinogen, fibrin can never be formed. So blood will fail to form a firm clot. There may be a primary hemostatic plug which may be formed if platelet numbers are insufficiency, but the stable hemostatic plug will not be formed and result in severe hemorrhage. Now, another important deficiency would be the deficiency of prothrombin. And this condition, factor 2 deficiency, it is called as hypoprothrombemia. That is deficiency of factor number 2. If it is less in amount, it causes hypoprothrombemia, and this can occur in two important conditions. One is liver. As I told you, any type of most of the clotting factors are synthesized within the liver, and any type of liver disease. It may be liver cirrhosis, it may be malignancy of the liver, it may be, and even there may be some condition wherein the liver is fatty, fibrinoid, etc. Then what happens is the prothrombin bleeding, clotting time, particularly prothrombin time. These are certain tests which are performed in the lab. Blood sample is collected, bleeding time, clotting time, and factor 2 deficiency is evaluated, and that is called as prothrombin time. So the prothrombin time becomes very long, abnormally prolonged, and therefore excessive bleeding or hemorrhage can occur. So liver disease may be responsible for the any type of liver disease, liver malignancy, hepatitis, cirrhosis, malignancy of liver, they can be responsible for lack or insufficient amount of prothrombin. In addition, sometimes jaundice is there, hepatitis or jaundice. And if it is obstructive jaundice, there is a particular, another vitamin, calcium is required at every step. But a vitamin also is required at required for synthesis of prothrombin, and that is vitamin K. In fact, vitamin K is synthesized by bacteria in our intestine, and the vitamin K which is synthesized, it is absorbed. So in obstructive jaundice, the vitamin K factor, which is essential for synthesis of prothrombin, it cannot be absorbed properly. And why this happens? There is some obstruction in the bile duct, and there may be insufficient amount of bile may be there. So due to insufficiency of bile salts, vitamin K is not absorbed, and due to lack of vitamin K synthesis, prothrombin synthesis is not there. And factor 2, factor 7, factor 9, and factor 10, all these factors may be less in amount, and this can cause hypoprothrombemia. Factor vitamin K deficiency in obstructive jaundice can cause, or failure of vitamin K to be absorbed due to absence of bile salts. This will result in lack of synthesis or insufficient synthesis of prothrombin, and this condition will result in factor 2 deficiency called as hypoprothrombemia. Then we come to factor 8 deficiency, factor 9 deficiency. Factor A deficiency, as I've already told you earlier, causes hemophilia A. And in hemophilia A, it is this particular condition or disease. It was discovered in a royal family, the Queen in Spain, and it is caused by a sex-linked gene, recessive gene. That means the gene is recessive. The gene for normal clotting process is the dominant gene for factor 8 synthesis, but this particular gene for synthesis of factor 8 is present on the sex chromosome. And if a recessive abnormal allele is present, and it is quite common in males, all sex-linked diseases are common in males, and therefore hemophilia A is quite common in females, males, sorry. And here the blood takes a very long time, even after a very minor injury, to clot. And there is delayed clotting of blood due to factor 8 deficiency. And there is a tendency of what is the manifestation? It manifests in bleeding disorders. That means tendency to bleed excessively, severely, even after trivial or minor injury. And here the clotting time becomes abnormally prolonged. And what is the treatment? We can provide factor 8 concentrates. So even transfusion of blood can be given, or transfusion of plasma, which is rich in factor 8, or factor 8 concentrate can be given. Then factor 9 deficiency, of course, it is the anti-hemophilic factor B. So factor 9 deficiency leads to Christmas disease. And this is congenital. Again, this is congenital and results in delayed formation of fibrin. It results in delayed formation of fibrin. It is also caused, the gene for this is present on the sex chromosomes. Therefore, we say it is a sex-linked gene and it is recessive in nature. But here, in Christmas disease, the bleeding is not as severe as in classical hemophilia A. Lastly, there is something called as pseudo-hemophilia. Whenever there is insufficiency of factor number, deficiency of factor number 5 and 7, that is the labile factor and the stable factor together, then it leads to a clinical syndrome which is called as pseudo-hemophilia. This is a hemorrhagic condition, and here this hemorrhagic condition is due, there is bleeding disorder, but it is congenital. It results in delayed formation of fibrin network, and it is again, the gene is present on the sex chromosomes, and it is caused by a recessive gene. It is caused by a recessive gene, this particular disease. Pseudohemophilia. The gene is present on the autosome. It is a recessive allele. It's not extensive. The hemorrhage is not extensive, but it simulates, or it copies or imitates the condition of hemorrhage caused due to hemophilia. Thank you very much. I thank you all for your kind attention. I am now ready to take any questions the students may have. I'm ready to take questions if anyone has any questions. Any students have any questions? Sir? Sir, Milind sir? Hello, sir. I've completed my topic. I'm ready to take any questions if there are any questions of the students. Okay. Okay, I'll wind up. I'll end the session. Thank you, sir. Sir? Can you send me the link to the sir told me that you'll be sending me the invitation letter, the thank you letter, and the link